Swayam Prabha. Digital India. Educated India.
So, but in any case, if you are aware of someone who has not participated in the thing and you still can talk to them, it would be nice that you have a chance to convince them that it may still be fun and interesting to have them participate in that. Okay, so that would be one thing, but today will not be assignments for papers. What I want to comment now is a little bit this last part of the course. In particular, the last day, that, as I told you, I want to do three things during that day. First of all, I want to have the presentations of your IM projects, and I will return to that in a moment. Then I will have to have a conversation with you on what have been your reactions and your kind of take-home lessons of whatever you have heard and discussed during these days that we had the course. And finally, we'll have to have a real reception or party, so we get together and kind of talk friendly on different aspects of your life here and everything. Okay, so some hints about these IM projects. So this morning I sent you an email, I don't know if you had a chance to see it, with this famous Romeo and Juliet thing, okay? So obviously I don't want all of you to do something similar, but it's just to see the type of projects that you are welcome and kind of challenged to present and to develop the last day of our meeting of our course. So maybe you can use that as a kind of pattern, but really I would encourage you to go to the IM webpage, take a look to different projects. There are hundreds of different projects, and you more or less figure out a project of this sort, okay? So in the project, basically what you have to structure, and I would like each of you to prepare a little presentation, maybe a few slides, you don't need to have a bunch of slides there, is about the concept, what would you pretend to do, what would be the parts, not an exhaustive list of parts, but just at least an indication of the parts, the activities that you will require for making that project a reality, and what you think would be the, what you may call the socio-economic consequences of that project, okay? So obviously you have the project on Romeo and Juliet, the socio-economic consequences would be zero, but you know, maybe you decide to produce a bacterium able to convert plastics into, I don't know, a super expensive drug, or maybe you want to develop a biosensor for doing this or doing that. I mean, you have many different projects, okay? So one thing that we have to see tomorrow is how many of you really sign up for this activity, because the last day will be a very busy day and we have to control the time and everything. But I would like each of you to have your little minutes just to share what type of inspiration this course has elicited in your view of things and what type of crazy ideas you would be willing to share with the others, okay? So ideas, by saying crazy, mean that they have to be out of the box, but they cannot be absurd, right? So like say, I'm going to make, I don't know, a bacterium that, you know, I jump on top of the bacteria and go to the moon. I mean, that would be completely ridiculous, right? But if you propose something that makes some sense, you know, like, I don't know, one of the projects that I may mention in the seminar this Saturday is that, you know, I make a tree that when it grows produces a house, and then in the house, you know, we have an elevator that is not made with metal, it's made by some osmotic pressures. I don't know, I mean, something that in first sight may look a bit crazy, but it's not completely absurd, okay? It can be a bit absurd, but not completely absurd, right? Okay, so this is the type of challenges. So please think about it, think whether you want to participate in this. It will not be a contest, there will not be prizes, but you know, it will be nice to, and fun to have this type of dialogue on crazy things that can be done. So please tomorrow think about it, think whether you are willing and motivated to get into this type of final discussion, and let me know, because I have to organize a bit the schedule and so on, and make sure that everyone has the time to do it, okay? And then we'll have this little reception and so on. And also please, and this is very important for me, do express your feedback, you know? And I would ask you one by one, the ones that are here the last day, what do you think of this course? You may say, well, this course has been a pain in the neck, and I would never think on synthetic biology for the rest of my life. That's fine, that's perfectly fine. But I want to have a kind of overview of what is your reaction, to what degree what you have heard these days is completely novel in your CV or in your say, curriculum, to what degree you have heard new things, to what degree, I mean, these type of things, I'm very, very interested, and so on, and because as I told you, I'm trying to put together a textbook on synthetic biology, and I would like to have your early feedback to see whether, you know, it would be a good idea or something that is not reasonable. So you told me yesterday that now the textbooks are mostly PDFs, right? That you don't buy the thing, right? So I don't pretend to be a millionaire with these sales, but I think that a book of this type could really help to shape a community of people all over the world. I mean, not just because of my book, but you know, sometimes one discipline gets a structure because someone writes a book, you know, and that can be what you have in mind, or it might not be like that, but you know, for instance, the whole field of systems biology, there is a super famous book that was like the founding, say, action or the founding occurrence in systems biology. You know what book is that? Uri Alon? Yeah, that's okay. So Uri Alon is this gentleman in the Weizmann Institute in Israel. And before this book, systems biology was a kind of word that people use here and there, so, but no one really had a concept of systems biology. And he wrote a book, this systems biology, it's called Introduction to Systems Biology or something. Well, anyway, and then since then, this book has become a reference. So everyone that gets into systems biology, sooner or later has to go across this book, because it's where all the nomenclature is explained, all the basic concepts, all the circuits, the feedback loop, all these things are explained in detail, and this is something important. So I would like to at least to try to do something like that, and that may be useful as well, because there are many reports on synthetic biology, what is synthetic biology, what is not, the jargon, everyone uses different words and everything, but at the end I think that it's good to have a reference to clarify what is what, and mostly the basic concepts that we have discussed the first days. I think it's important what is a part, what is a standard, what are the requirements for the standards, what is a circuit. I mean, all these things require some formalisms and some standardization, also in semantic terms, that I believe a book can help to put together. Okay, so please remember tomorrow, whether you come or to tell your friends to or your colleagues to come, you tell me, I will go around whether you want to participate in this IEN thing, and then we will have a final nice and fun and entertaining finale of the whole course. Okay, so I stop now, and then we start with the presentations. So who wants to start? Okay, so maybe we should start by you, Adita, because you were supposed to talk yesterday. Okay, please go ahead.
All right. So, So physical interactions of gram-negative bacteria, and in this case, Pseudomonas petita, are mainly mediated through these specialized structures that are present on their outer membrane. These structures are pili, flagella, fimbriae, and some other extrapolymeric substances. And these sort of outer membrane structures are involved in cellular motion and adhesion. So in this image here, you can see Pseudomonas petita strain KT2440, and this strain has all these outer membrane structures. Now, this study used Pseudomonas petita strain KT2440 as a biological chassis, and the reason as to why this was done is mainly because it's a non-pathogenic variant of Pseudomonas, and it also withstands oxidative stress. This is mainly due to the fact that its metabolic architecture is such that it produces a large amount of NADPH molecules, and this microorganism also possesses a high tolerance to organic solvents. Now, all right. So, as in natural environments, the Pseudomonas petitas, motility and all these outer membrane structures are very beneficial. But in terms of like a biotechnological application or a synthetic biology application, it's not exactly the greatest thing. That's mainly because its function and production is energetically expensive. And this energy could, potentially be directed elsewhere. And these extrapolymeric substances in the Pseudomonas petita have also been shown to cause some biofouling in reactors. Now, so over the years, what scientists have done is they've sort of deleted certain segments of the genome of Pseudomonas petita. And I think this was mentioned in yesterday's, one of yesterday's presentations, that scientists deleted about 4.7% of the genome of the Pseudomonas petita KT2440. And they deleted all those outer membrane structures and some burdensome segments. And this gave rise to the Pseudomonas petita strain EM371. Now, there are very key characteristics of this strain. And the main one is that, this strain cannot build biofilms. That's mainly because its outer surface is such that, there isn't any place for cell-to-cell interactions or any adhesions to target molecules or surfaces. And so because this naked cell over here, it has a very sort of smooth surface all around it, it basically ensures an accessible overlay that facilitates the display and functionality of adhesins. And this strain of Pseudomonas petita also displays a better tolerance to stress. Now, this paper basically talks about engineering tropism, which is, in this case, it's essentially a sort of a preferential or conditional attachment to a target surface. So in short, what this paper is doing is it's projecting a nanobody from the surface of the Pseudomonas petita, the naked Pseudomonas petita variant, and this nanobody is attaching to a molecular target, which is coated on a surface. Now, so this has been done through a couple of different ways. So the study adopts these VHH nanobodies, and as we'd seen before, they're camelid antibodies that, they're a single polypeptide, while, they still, they're single polypeptides that still have full recognition of the antigen epitopes. Now, these VHH nanobodies are facilitators for the interaction between the antibody and the antigen. And the VFib1 is what we're using here, and the antibody we're using, and this recognizes human fibrinogen. And it binds to it. So this VHH sequence, as you can see in this image, is sort of fused to this intamin carrier. Now, intamin is the beta intamin carrier. So intamin is basically a sort of gene that's found, or protein that's found in E. coli, that certain virulent or pathogenic strains of E. coli, which help in displaying or projections on the surface of the cell. And this is essentially what's taking the VFib1 protein to it's displaying it on the surface of the cell. Now, this entire system, it's being induced with a 3-methylbenzoate xyl-SPM system. And this has all been put into a P-SEVA 238 vector, and we're using this to transform the cells. Now, coming to the expression of the VHH fib in Pseudomonas petita, so we can see that in the presence of induction over here, which is in the presence of 3-methylbenzoate, you're going to see a very dark sort of band over here, and that essentially indicates the production of this intamin-fused VHH fib1. Now, notice that you can sort of see fainter bands that indicate sort of a minor product as well. So basically what we found that, when we pre-treat these samples with urea, this minor product is just, it's not there at all. It's virtually just disappearing. And the reason, so basically this gave us so because of this, the study drew the conclusion that there was only partial unfolding of the protein, which is sort of why we're getting this minor product over here. And another thing that this Western blot sort of showed is it showed how this expression was embedded in the metabolic network. So to test that, what they did is they took a anti-GRO-EL antibody that's used as like a general stress reporter. And GROW-EL, as I think has been mentioned before in one of the other presentations, it's a protein complex that helps in the folding of other proteins. And this essentially showed, very like constant or like very like similar sort of sizes of the GROW-EL, or they're all in like the same straight line. And that essentially implied that, this sort of VHH fib expression is not harmful to or detrimental to Pseudomonas putida in any way. Now, we had said that, this sort of nanobody is something that we want to display on the surface of the cell, right? And this study essentially was checking to make sure that the intimate carrier actually did make it, produced on the surface. Now, to check that, what the study did is that they transformed these Pseudomonas putida variants, both of them, with a protein called TRX-G6-VHH. Now, this protein exists only inside the cytoplasm and is not excreted out in any way whatsoever. And so note that, this had an E tag over here, and I'll go back actually, a couple slides. Yeah, okay, again, this sort of VHH Fib1 also was flanked by an E and a MIG tag. Now, coming back to this, again, so these cells were treated with a non-lethal concentration of trypsin. Now, trypsin is a protease that essentially, cleaves the proteins in, its general vicinity. So the thing is, with trypsin, it cannot actually enter the bacterial cell. And so all the sort of proteins that were displayed on the surface or in the surrounding regions where trypsin was introduced, all those proteins were cleaved. Now, coming to the Western blot that we can actually see here. So over here, we can see that, in the presence of induction and in the absence of trypsin, you have VHH Fib1 being expressed. And again, in the case of TRX, in the presence of induction, in the presence and absence of trypsin, you're going to see the presence of this protein. And that's because this protein exists only inside the cell, inside the cytoplasm, and it doesn't exit. Now, in the presence of induction and in the presence of trypsin, the IVHH Fib1 shows virtually no sort of expression. I mean, there is a very faint line that you can see over here and down here as well. So the theory sort of posited this hypothesis that the reason that there's, this residual fraction of this fusion that's, unaffected by the protease treatment is mainly because there's an intermediate step in the display of these proteins where the E tag, which is what the protease trypsin cleaves, it's not fully, shown, and the trypsin can't actually cleave it. So that's sort of essentially proving the surface display of the IVHH Fib1 in the Pseudomonas putida strains. Now, now that we've, shown all this, we had to make sure that the bacteria the bacterial nanobodies actually attached to the target surface that you wanted. And because it's bacteria that we're checking, right? You need like very precise attachment metrics of these bacteria onto like the target surface. And so this was done using a quartz crystal microbalance. It's essentially a very, very sensitive mass sensor. And the way it works is by sensing the changes in small amounts of mass on the electrode surface. Basically what that does is it changes the oscillation frequency of the crystal. That's measured by delta F, and that's actually what's characterized. Now the quartz crystal microbalance also allows for continuous measurements and of the interaction between the antibody and the antigen, and it also provides kinetic information on the process and the resulting surface coverage. Now, okay, so essentially what this entire sort of graph shows is that once fibrinogen is added to the cell surface, to the target surface, there's sort of a decrease in delta F, which is some mass has been added to the surface of the electrode. Now, which is sort of leading to this sharp drop in oscillation frequency. change, sorry. Now, once it stabilizes, the bacteria are added, and that leads to another drop in the oscillation frequency, because that essentially indicates the binding of the antibody present on the surface of the bacteria to the antigen, and that in this case is fibrinogen. So as we can see over here, let's just go straight to the C part. As we can see, after the addition of cells, we're going to see a sharp drop in the sharp change in oscillation frequency in both the Pseudomonas petita strains. Now note that you can see sort of like a larger drop in Pseudomonas petita KT2440 over EM371. So it's a pretty significant drop. And now coming to, okay, so one more thing was mentioned that, the QCM could also check the kinetics of the interaction. And it was found that the EM371 strain had a higher rate of attachment to the fibrinogen on the target surface. So I think it was 0.03 per minute, as compared to 0.003 per minute in the case of Pseudomonas petita KT2440. Now, so on this quartz crystal microbalance, they took like some scanning electron microscopy images, and this was the result that we could see. So in the presence of no IVHH Fib1, there was essentially no antibody that could attach to the antigen, and we don't actually see any bacteria on the image. Now, we had sort of mentioned that in the previous slide that in KT2440 strain, in the presence of the nanobody, that there was a sharper drop in the oscillation frequency, as in the case observed with 371. So that resulted in sort of like this theory that was posited, and essentially saying that while that it's less efficient for the nanobody to fibrinogen attraction over there, or attachment, there's sort of like, again, one more thing to remember is that the KT2440 has the outer membrane structures. So once this nanobody attaches to the molecular target, all the other bacteria sort of bunch up, and there's like a lot of cell-to-cell interactions, and there's sort of like a biofilm formation as well. So they just sort of like cluster around and aggregate in a certain specific area. But in the EM371 strain over here, we noticed that there's like a monolayer that's formed of bacteria because, as we mentioned before, there's no biofilm formation that happens in this strain. And you can see this like large monolayer that's sort of resulted, and it's very spread out. So that's pretty much concluding the summary of the paper. Now coming to the critical analysis of it, so I'll go back to the slide over here. Right. So here when we saw the IVHH Fib1 sort of being expressed, and we saw this minor product as well, right? Oh, sorry. Yep, sorry. Over here. So there was this sort of minor product that you could sort of see over here, and the reason I'd said, or the study had said, was that it's present because there's sort of this intermediate step that takes place where the E tag is not fully exposed and the protease can't actually cleave it. But the study never actually went on to show what this intermediate step was, nor did it posit like any sort of experimental like validation for this theory. Now, that was sort of the cons of it, I suppose. Now, I can actually see the overall application of this monolayer that you're forming over here. It's like it's a wide-ranging applications that you have. It's like, let's suppose in plastic degradation, like plastic degrading bacteria, you could probably, this basically would maximize the area of, sort of maximize the amount of, plastic that can be degraded using like the least amount of bacteria possible. Because in KT2440, the bacteria sort of aggregate and form like multiple layers that don't necessarily have any, say in the production or the degradation of the plastic. So that's pretty much it. Thank you.
Okay, thank you. Well, thank you. It was a very good job. So just in connection to what you mentioned on what that you don't say applications, and you introduce an interesting angle to the whole thing, because you know in synthetic biology you have to distinguish between models and games. I mentioned that on passing in one of my talks. So obviously this is a game. This is not a model, it's not an application, right? So in a game, what you do is that you take the different components of the system out of their context, play with them, see how they behave, and then you try to produce rules for functioning, okay? So in this case, the target obviously is not plastic, but this tells us that it is possible to do these things. So in a project that is currently undergoing on plastic biodegradation, what we have is a number of peptides that have been developed to stick to polyethylene. Okay. And then on the other hand, we have some bacteria that secrete some proteins that appear to have some activity on polyethylene. Okay. So the kind of real application is the one where you bring this bacteria in contact with the polyethylene, but that's the application, that's not the kind of basic endeavor that we have here. So remember what we discussed yesterday about this proof of concept. So I think this is the proof of concept that you can use synthetic adhesives to adhere bacteria specifically to a surface of your choice. And then what comes after is a different story, okay? Okay, well, thank you very much. Good job. All right, so next speaker.
Okay, so hi everyone. I am Ashna and I am going to be giving a short review on this paper, measuring the activity of BioBrick promoters using an in vivo reference standard. So first I want to start with a little background on this paper. So we know that as we've learned in previous classes, standardization is a crucial thing for engineering complex synthetic biological systems. However, as we know, biology is very complex and this makes it difficult to standardize. So one very successful step forward in this has been the BioBrick standard biological parts. However, the main aspect for standardization that BioBricks use is limited to the physical assembly of the individual parts. And so this paper explores another possible standardization aspect that we can implement. So the first step for characterizing standard biological promoters was to choose a particular property that would be best for reusing the part, and that would be most relevant to biological engineers. So since we know that promoters are primarily used for initiating transcription, the rate of transcription initiation was taken as a property. And they've defined promoter clearance activity as the specific property they want to quantify in this paper. So promoter activity is defined as the number of RNA polymerase molecules that clear the final base pair of the promoter and continue along the DNA as an elongation complex. So promoter activity has been measured in the unit of polymerases per second, or POPs, as we've learned earlier. However, as mentioned earlier, directly measuring POPs in vivo is a very challenging task. So this paper utilizes the addition of the promoter upstream to the coding sequence for green fluorescent proteins, or GFPs, to correlate the POPs value to GFP synthesis rates. And they've done this using a previously described differential function. So this is the differential function for absolute activity of the promoter. So they have different parameters mentioned here. So when running tests, we know that there are going to be variabilities due to the experimental conditions and the equipment, etc. So the sensitivity of the GFP values was basically explored under different measurement conditions and instruments. And so the first thing they did was they tested this across two promoters. So GFP synthesis rate was basically estimated by the change in an arbitrary fluorescent unit, as they've defined, per absorbance over a 1 hour long period in the log phase growth. So there were several variations in environmental conditions that they've considered for analysis, and I've mentioned those over there. So they found that cell strain was one of the conditions that affected the value of the GFP synthesis by around a factor of twofold. But it was also found to be insensitive to the carbon source and the temperature. And the plasmid copy number and antibiotic resistance marker also had some sensitivity when the experiments were run. So all of these are summarized in these two graphs here. So graph A basically shows the difference in the two promoters that they've considered. So the left one, the white one, is the J23101 promoter, and the gray one is a J23150 promoter. So as you can see, there is a very large extensive difference between the white and the gray values in the graph A. So graph B basically shows the relative activity of J23150 with respect to J23101. So as we can see, the value, the variability coefficient is a lot lesser for graph B than graph A, which shows that calculating a relative activity is probably a better way to standardize this value. So again, as we saw in the previous two slides, the activity of promoters measured across the different conditions was correlated. So we're defining a new property here called relative promoter activity. And we're considering a standard of BBA J23101. And this has a big advantage because many of the parameters that are affected by many environmental parameters are going to sort of cancel out when calculating the relative value. So these are the equations that they've used, and they've basically taken 4 different assumptions to simplify the equation. So the first one was that they've considered the GFP expressed from the test promoter and the reference standard promoter to have an equivalent maturation rate. So which basically equates to the A values in the second equation. The second assumption that they took was each promoter was estimated to have the same average copy number. So the N values in the equation. The third one was that the mRNA degradation rates were also assumed to be equal across the promoters, and that is signified by gamma. And the last assumption that they used was they assumed the translation rates of the immature GFP and mRNA to be equal, which is signified by the rho value. So they've taken another additional simplification and they've obtained a final equation. So this is the final equation that they've got correlating the relative activity to the GFP synthesis rate. So so the next factor that might come into play is variations when tests are run across different laboratories. Because each lab is going to have its own distinct set of protocol, there might be a difference when these tests are run in different places. So to basically consider this as a factor, a reference set of around four promoters was sent to 6 different labs, and these tests were considered these tests were run with some general rules, so that we can see if the results are correlated or not. So this is the graph for this is the plot for the tests being run in the different laboratories. So as you can see, there's a reasonable correlation between the values of the relative promoter units across all the labs. And so basically from this we can conclude that this is a good this is like a good metric to standardize for biological parts. Then we come to basically, so since they've established that the system is a good metric to utilize, they want to basically have a more widespread adoption of this. And for this, they've basically utilized the relative activities of 7 different representative promoters from the registry of standard biological parts. So these promoters, show the next slide. So these are the 7 promoters that they've used for the standards. And 5 of these, the first 5, are from the constitutive promoters. And the 6th one is a TET repressor activated one. And the 7th one is a LAC repressor activated one. So to further support community-based standardized measurement, they've, the researchers and colleagues in the paper have basically created a first-generation measurement kit for characterizing the relative promoter activities in RPA values. So coming to my critical analysis, I think, so as we know, like standardizing is a very essential part of creating biological systems, as mentioned earlier. And it's always important to look at new methods of standardization, and basically characterize your parts and systems better. So I think this paper had a very novel approach to this, because before this paper, mainly biological parts were characterized by the physical assembly of the part itself. So I thought this was a very unique approach to this. Yeah.
Okay, thank you.
I think that's it. Thank you.
Don't rush, don't rush. Okay, so, well, I think that this is an interesting paper, as you mentioned, because it was the first attempt to put some real numbers behind. However, the last part that you mentioned a bit in a rush, I think is very important, is this interoperability among different laboratories and so on. And I don't know whether the paper stops there, but you know that after this paper there has been some inter-laboratory studies on how the different promoters work and everything. Okay. And you get these big variations, okay? So I think that there's still a big issue on the way we measure experimentally different parameters. Right. Because there are big, I mean, we know that the weakest promoter is weak and the biggest is strong, but we don't understand.
Quantifying that is
exactly,
a difficult task.
Exactly, so the parameterization of these promoters is still a big issue. Okay? Right. So you think that this is something that in the future may improve, or we have to live with this degree of variability in biological parts?
I think that we might be able to move a few steps forward in this, getting more precise results. But I'm not very sure how exactly that would work, but I do think that there might be some parameter that may not have been considered yet, that might have better correlation, that could be better considered for standardization.
Yeah. Okay, very good. Any comments? Any? No? Okay, well, thank you. Thank you very much. And we move to the next speaker. Yes.
Hi everybody. Today I'll be discussing a paper titled New Transposon Tools Tailored for Metabolic Engineering of Gram-Negative Microbial Cell Factories. So basically in this paper they have used a transposon tool for doing genomic integrations in gram-negative bacteria. So yeah, so here we go. So coming to the design and characterization of this mini TN5 based vectors. So they have used this PBAM vector. So this is all these 3 modules are the derivatives of the native PBAM vectors. So the coming to the components of this vector, so it basically has an antibiotic resistance marker, and it also has a TNPA gene which codes for a transposase. And it also has RK6, which is the origin of replication, which is the pi-dependent one. And also they have this RET, which is responsible for the transfer. Also they have something called MEs, so they are mosaic end sequences, which is recognized by the transposase. So basically these 3 modules are just differing in 3 antibiotic resistance markers. So PBAMD 2 is for kanamycin, and 4 is representing streptomycin, and 6 is for gentamicin. So they also have multiple cloning sites, which is having a lot of restriction enzyme sites, which can be used for inserting our DNA cargoes into the genome of the microorganism of interest. So basically, they did the validation of this transposon tools in, of course, Putida KT2440. So the assay was done using the triparental mating, where they have this donor cell, recipient cells, helper strains, and also the transposon, the strain which has this, which expresses this transposase. So basically, the frequency of transposition event was estimated by determining the number of antibiotic resistant colonies. And in order to differentiate between transposition event and a non-specific plasmid integration, basically they did 2 simple things. One was just testing the growth of this on the growth on ampicillin, because this vector PBAMD had this ampicillin resistant. If it grows on it, it means that it still has this vector. And other method was by doing a colony PCR. So where they, if they use these primers, and if they get a 225 base pair amplicon, it is representing the presence of plasmid. And it also, if we also use another primer pair, we get amplicon of 665 base pair. It represents that the plasmid is still there. So there was a 4.2% potential transconjugance obtained with this vectors, which was resulted from the plasmid co-integration event. So this was working quite well. And so they also did a sequential insertion. So when they have to insert 2-3 things sequentially, this mini TN5 modules also worked pretty good. So first they took a KT strain, and then they inserted the kanamycin module in it, and then they also sequentially or serially inserted other genes of this antibiotic resistance, and they were able to serially insert all 3 antibiotic resistance determinants, and they also assessed the possible sequence preference in the target DNA during the insertion. So what is the sequence preference while insertion? They also determined this, and they also could track where it exactly goes and inserts by doing arbitrary PCR with that MEO sequence. So they have tracked where and all the insertion has happened in that serially inserted things. So basically they could track most of the insertion sites except for 2 cases. One was that they couldn't predict, it's this one, the KTG1B. So they had this two hypothetical proteins which were 97% sequence similar. So they couldn't track which one was it. And then the other case was in the position where it codes for a gene LAP-A surface adhesion protein. There were a lot of internal repeats in it, so they weren't able to identify the exact insertion site. So they were able to successfully do the integration and all in a random manner. It was not a specific one. And they also used the system for PHB production, where they inserted, as sir already discussed about this, where for PHB, which is a bioplastic which has a lot of applications, it needs 3 essential genes, thiolase, reductase and a synthase. So they have taken the genes from Cupriabides decatur and they have inserted into the genome of a E. coli mutant. So basically they had a E. coli which is lacking this gene, which is called phosphotransacetylase. So this is a gene which is responsible for the production of acetate. So this acetate is a byproduct in the production of PHB. So by knocking out this gene, they could get very minimal levels of acetate, so that the flux is directed more towards the accumulation of the biopolymer. And they also have shown this image, the PHP content. So when there is insertion at different site, the expression levels was varying. This is important because you could see the high level of expression or production of this PHP in some insertion, and then there is very less in other case. So this is really important, and one has to have a account of it and go which one is the best for them. So they can just take that. And yeah, so they also have different strains. So this strain BW25113 is a wild type strain, which is without any modification, and the JW229 is the acetate knockout strain. And then the next one was the E. coli acetate knockout with the PHB production genes, and then that was expressed in a plasmid. The last strain, which is TA293, was the best producer, which was the knockout of acetate plus this PHB genes, which was genomically integrated using the TN5 strain. So when you looked at the activity of the PHA enzyme, which was the first enzyme which is responsible for the production of PHB. So in first two strains, there were very minimal levels of PHA activity, but then the strain which had this PHB genes in the plasmid had a much higher activity of PHA, and also the strain which was genomically integrated showed a higher level of activity of PHA enzyme. And also when it comes to PHB content, only the enzyme activity is not enough. So when it comes to production, so as you can see, the plasmid-based system showed the highest PHB content, but the insertion also showed about around 62% production when compared to the plasmid-based system. And one more important thing is the highest producer, that is TA2293, which had the highest level of PHB production. The insertion site was at a region which encodes YKGH gene. So this gene is an open reading frame encoding a inner membrane protein. So the insertion site is very critical when it comes to the production. So one has to keep an account for that. And yeah, so the conclusions are, so they have developed a set of TN5 derived vectors that are standardized and that can be used to engineer a wide range of gram-negative bacteria. And then the possibility of serial insertion is also possible, and it can be done in a combinatorial fashion. And also, the other thing is the genetic context of each integration will definitely result in a different regulatory pattern at the transcription level, and the user could choose the best one among the library of insertions. So these are the conclusions, and yeah.
And the critical appraisal?
Yeah, so this is really a great tool for when we have to do insertion of any gene of interest for a wide range of application. So I myself have used this tool for integrating genes responsible for biosurfactant production in Pseudomonas, but that's a different species. But one thing with this, it is random, not a specific one. So again, they have come up with a solution, a much better version, TN7, right, which is much more specific. So that's a way to go, and it's good.
Okay, thank you. Okay, questions, comments? No? Yes?
So, you said the genetic context causes the changes in the levels of expression of the PHP information, right? Is that based on the promoters tracking the sequence of the insertion? When you insert that sequence, which is like producing the PHP, in the genome present, there might be some promoters which are tracking the insertions. So is the strength of that promoter causing the?
Well, I think it's not only the effect of promoter; it's the combined cumulative effect of lot of regulatory things that are happening.
Yeah.
Is that right? Yeah.
I fact, that's a good question, because tomorrow we will analyze exactly that point in great detail, okay? Because, you know, in the context of to what degree you can do top-down genetic engineering, I mean engineering and complete predictability, and to what extent we can play with variability also to optimize a given device. This is something that I will discuss tomorrow in great detail. But thank you very much. Okay, so we have the last speaker today. Yes.
Hi everyone, I am Muthu Arnachalam. I am from first year Masters in Bioprocess Engineering. I am going to briefly talk about the topic of versioning biological cells for trustworthy cell engineering done by Jonathan et al. And whenever we think about barcodes, we can see them daily in commercial products. They are unique representation of certain digits which are used to identify certain products and their information from the databases. And in this paper, they are talking about how can we synthesize or create a DNA sequence which can be tagged to the genome of a modified bacterial strain, so that whenever we forget what kind of modification we perform on the strain, that time we can use the sequence to get the information about the modifications performed on the strain. So these are all the reasons that are required for barcoding the bacteria. First of all, traceability. Whenever a genetically modified organism is present in the environment, it may pose a threat, so that let us consider every genetically modified organism is barcoded. Then we can sequence the strain. Then using the barcode, we can identify what is the strain and what are all the modification it has from the database, so that we can also find the source also. Then we can charge on the responsible person. And reproducibility of the experiments. When we see some papers that are doing some experiment using the strains, if you want to reproduce means we can, I'll show here. This is a database generally. What happens means a user who is going to generate a strain first selects the strain he is going to operate on, and these are all the modifications he is performing on the strain. After some modification, he chose this as a potential strain, so he asked the system to generate a particular barcode. So it gives some sequence of DNA. Using that sequence, we are chemically synthesizing the sequence and barcoding inside this bacteria. If we do this process, then the bacteria can be barcoded with that sequence, so we can identify it from the database. And also from this database, we can share our information with our collaborators, as well as they can also modify some experiments that are performing on them from the database. And this is the process. After the system generates that particular sequence of nucleotide, we can inject them into the cells using recombinant DNA technology like homologous recombination and CRISPR-Cas9 technology, which we have already seen in this class. And we have to make sure that the barcode survives in the cell after a long time. Generally what happens means there is no selection procedure for identifying if a strain has been barcoded or not, other than sequencing it. So it's very difficult. If we think that there are two types of strain, one is barcoded and another is unbarcoded in our solution, then if we grow them for a lot of generation, if the barcode causes some errors in metabolism, the barcoded strain may lose upon subsequent generation. So what we do means we have to check before using the sequence as barcode. So a sequence of the barcode should not be interfering with the growth. We can identify it by growing the strain as well as the wild type strain in different conditions. And the barcode should not code for protein, else it might interact with other proteins on the cell, and it should not interfere with the regulation that is already present in the cell, like interference with promoters or distant regulatory sites. and the stability of the sequence also matters. Like when we do the recombination process generally generates double stranded breaks. This is the reason which the cell recognizes some thread and follows the homologous recombination. Here, this is what we want, homologous recombination. When there is a double stranded break that is present, it recruits some enzymes. So if we have some repair template, then our sequence of interest, that is here the barcoded region, will be integrated into the genome. But sometimes non-homologous end joining, this process also happens. Here what happens means the template might not be available at the site of action, so some errors in the sequence also takes place. This results in the death of the cell because it introduces non-random mutations and trips the ends that are present there are shortened. Also, here we can show a role to the scientific community so that they can understand what kind of works we are doing, and they might suggest some better ideas. So in case if we make some errors in our experiment, it might cause some losses in our funds. And finally, this paper just tells that we can make some non-functional piece of DNA and use it to identify what kind of modification that are performed in the strain without affecting its function. And I found this thing interesting that in case if we forget to label a sequence, label a strain that is present in the laboratory, and if you want to find what is the source, then we can sequence it and find the information from the database itself. And if there is another strategy, like we don't have to sequence the DNA and find it, like if we directly PCR it and see in a gel, it might form some patterns. If the technology is like that, we don't have to sequence it, and it might reduce time also. So I thought it might be a future alternative.
Okay, thank you. So, according to the discussion we had the other day on containment versus traceability, what would be your opinion? I mean, what is better, to have something that is contained, or is something that is traceable, as in the case of barcodes? Or you think that both are good, or both are bad, or we have to look for a different thing?
So containment, in case if that is potentially dangerous to others, it should be contained. And you're asking about we have to contain, or is it present outside?
Well, you remember I don't know if you were in the class, we had this discussion of whether once you have a recombinant construct, a synthetic biology agent, you want to make sure that it does not spread beyond the limits that we are interested, or, as that is very difficult or eventually impossible, whether we should be happy enough having an unique identifier. What is your opinion?
So I think most of the strains that are used in the labs are not able to compete with natural microbes that are present outside. In case if it poses threat to the ecosystem, then we might have to find some strategies to kill that particular microbe by finding out its source and something like that. We can do it, but still it won't be possible to eliminate all the DNA that is present in the environment. It might transfer DNA to other organisms also. That is also a difficult thing.
Okay, so in principle you would still bet for containment.
Yes, sir.
Okay.
Traceability is better in case if we have some strains that are mixed up in a reaction. So if we want to isolate each and every strain, then we can streak it, and we may isolate every kind of strains. It may be possible in some scenarios where we have only one vial, but we have to save all the strains like that.
Yeah.
Hypothetically, in Jurassic Park, we also did.
Okay, very good, thank you.
Okay.
Any question? Any comment on that? Okay, great.
Thank you.
Okay, so I think that we had today a very interesting day, a bit intense in the first part, but well, I think that we are done with the presentation. I don't know, I mean, if we do it today, will be at least a strength of the time. I think we should do it tomorrow, if you don't mind. Okay. Is that all right? Okay, great. Yeah, because you know, as we had this new addition today, then, but we'll have time tomorrow, no problem, okay? Well, thank you. Okay, so so we wait for tomorrow's last presentation, but there will not be more presentations. So please tell your friend that she has to send me. She sent it already? Okay. Okay. Okay, very good. So I have to check my email and make sure that everything is there. And in any case, you are very welcome to go to the webpage and see that everything is in place and so on. Okay, great. So I will send you later today or tomorrow morning email to the whole group asking you to tell me, I mean, we don't have to say that in public, whether you are prepared and to really go into this IM-like thing or not. I mean, you don't answer, that means that you are not interested, but please, if you do, please say yes, I will be interested, and we'll make a real schedule on how we organize the last day, okay? Okay, so then in the interim period of time, what I would suggest is the following. So having 4 hours of class lectures every day, I think is too much for you and for me, okay? So I think that what we will do tomorrow will be to have this formal, say, two first hours, as we have done in the past. Then we have this little coffee break. Then we'll have a little recap, and I will tell you some stories that I have been doing during that period of time. And then the last hour, what I would suggest, and I hope you agree, is that you start taking the time to discuss this, I mean, to study and to work in these IM projects, okay? So you can do it here or somewhere else, but please, as we have scheduled this 4 hours per day, it would be nice that you use at least this last hour to concentrate in these IM projects. And if we do it here, and I am around, you have some doubt or something, I can help you, okay? So I think that we can do that. We can have the last hour like a study session. So you go through you please bring your computers and everything, and you can work here, and then we can make some progress with this development of individual projects, okay? Okay, let's do that. Excellent. So ladies and gentlemen, a pleasure as every day, and see you tomorrow, okay?